Welcome to KMR Advice. Today, we'll explore drug distribution and the factors that influence it. Lipid solubility and ionization are crucial for a drug's ability to cross cell membranes. Molecular size can limit distribution, as larger molecules may not easily penetrate tissues. Binding to plasma proteins affects how much of the drug is available to reach target sites, since only unbound drugs are active. The rate of blood flow determines how quickly a drug is delivered throughout the body. Finally, special barriers, such as the blood-brain barrier, can restrict drug access to certain regions. Let's delve into the body compartments. Divided into three types. First, the intracellular compartment is where many drugs exert their effects within cells. The cell membrane acts as a selective barrier, controlling entry and exit of substances. Second, the extracellular compartment, which includes the interstitial space, allowing drug diffusion between cells. Lastly, the vascular compartment, or intravascular space, contains the blood plasma, where drugs are initially distributed. The cell membrane and the endothelium of the capillary wall act as selective barriers, influencing drug movement between these compartments. Understanding these compartments is key to understanding drug distribution in the body. One compartment model. Certain drugs have characteristics that cause them to remain predominantly within the intravascular compartment. Drugs with high molecular weight cannot easily pass through capillary walls into tissues, keeping them within the blood plasma. Similarly, drugs that are highly bound to plasma proteins are restricted in their movement out of the vascular space because the protein-drug complexes are too large to cross capillary membranes. These properties lead to a one-compartment model, where the drug's distribution is primarily confined to the intravascular space. Let's explore the two-compartment model, focusing on extracellular distribution. In this model, drugs like quaternary ammonium compounds and mannitol are distributed between the intravascular compartment, where they circulate in the plasma, and the interstitial compartment, which is part of the extracellular space. These drugs do not enter the intracellular compartment, highlighting their extracellular activity. Now, let's examine the multi-compartmental model, which involves both extracellular and intracellular distribution. In this model, drugs like alcohol and phenytoin are distributed across the intravascular compartment, interstitial compartment, and intracellular compartment. This highlights their widespread distribution within the body, affecting various tissues and cells. Selective distribution refers to the tendency of certain substances to accumulate in specific tissues or organs. Firstly, iodide is selectively concentrated in the thyroid gland, where it plays a crucial role in the synthesis of thyroid hormones. Next, calcium is predominantly stored in bones, providing structural integrity and supporting various physiological functions. Lastly, tetracycline tends to accumulate in bones and teeth, especially during growth periods, which can lead to discoloration due to its affinity for calcium-rich areas. Aminoglycosides, such as streptomycin, exhibit selective distribution, primarily affecting the kidney and the vestibular system. In the kidneys, aminoglycosides can lead to nephrotoxicity due to their accumulation in renal tissues. In the vestibular system, these drugs may cause ototoxicity, impacting balance and hearing. Understanding this selective distribution is crucial for managing potential side effects and optimizing therapeutic outcomes. The apparent volume of distribution, denoted as VD, is a pharmacokinetic parameter that reflects the relationship between the dose of a drug and its concentration in the plasma. VD is calculated using the formula VD is equals to amount of drug in body divided by plasma concentration of drug. This parameter helps in understanding how extensively a drug is distributed throughout the body, relative to the plasma. A larger VD indicates extensive distribution into tissues, while a smaller VD suggests confinement to the plasma. Understanding VD is crucial for dosing regimens and predicting drug behavior in the body. The human body contains approximately 42 liters of total body water, distributed across various compartments. The intracellular compartment holds the majority, with a volume of 42 liters. Drugs like phenytoin and alcohol are distributed throughout this space, reflecting a VD of 42 liters. The extracellular compartment consists of interstitial and plasma spaces, totaling 12 to 14 liters. Aspirin, for example, has a VD within this range, indicating distribution primarily in these areas. The plasma compartment alone is 4 liters. Drugs such as heparin are confined mostly to the plasma, with a VD of 3 to 4 liters. Understanding these distributions helps in predicting drug behavior and optimizing dosing strategies. Digoxin has an apparent volume of distribution of approximately 500 liters. This high VD suggests that digoxin extensively distributes into body tissues, far beyond the total body water 42 liter. The large VD indicates that digoxin is highly bound to tissue proteins, particularly in the heart and skeletal muscles, which is why it is effective in treating heart conditions. Understanding the VD of drugs like digoxin is crucial for proper dosing and therapeutic monitoring. Thank you for watching. If you found this information helpful, please subscribe to KMR Advice for more insights into pharmacology and medical education. See you in the next video.